Welcome to Tiny Show and Tell Us, the bonus series where you write in with your favorite science news or factoid, we read your email aloud, and then we dive deeper. I'm Sam Jones. I still have a cold, and I'm here with my co-host, Deboki Chakravarti.
Hi, Sam. So last time we did this, we talked about farting galaxies, and we talked about broadly neutralizing antibodies.
We did.
We talked about it all. So for listeners, if you want to take us on a journey from, one end of the universe to the other, all you have to do is email us. You can send us a message at tinymatters@acs.org, or you can fill out the form linked in the episode description. Sam, I think it's your turn to go first.
It is indeed. My tiny show and tell us today is from listener AJ. So AJ writes, re-habituation of disgust. And so, sorry, I'm cutting in already, but I believe it was at the end of an episode. Mm-hmm. Maybe it was part of a tiny show and tell.
Yeah, I think it was my tiny show and tell.
It was. Right.
Yeah.
We were talking about disgust, but then we sort of went off on a tangent about disgust, things that we find disgusting.
Right.
We were talking about having babies and how there were certain things that we thought before having kids, we thought we would find really disgusting that actually didn't bother us as much.
Yeah, because I think the work was about how people respond to pictures of diapers and poopy diapers.
Oh, right.
Yeah.
Yeah.
Yeah, so that was the habituation of disgust. And so we definitely ended up probably going on a tangent about the things that we find gross.
Oh, for sure. Yeah, we're good at tangents. But anyway, this tiny show and tell us is sort of linked to that. So I just want to clarify that. Okay, here we go. So AJ wrote, re-habituation of disgust. Hi, Tiny Matters team. I work in a surgical pathology grossing lab where our job is to create a gross description of submitted tissues for color, size, texture, if there is a lesion on the skin, etc. When I first started as a student, I would get a bit grossed out. But now it's gotten to the point where if I see an eye or partial face resection on the pathologist assistant bench or the placentas being washed before their formalin soak, I don't mind at all. I will say the one thing that still squicks me out, which I didn't realize squicks me out was a term. That's a word. It's great. I'm going to use it. It still squicks me out is dermal cysts that squidge out their insides when you dissect them. I thought you might appreciate an anecdote from a medical person who's not a parent. AJ, we very much appreciate this anecdote.
Yeah, the range of things that you're dealing with on a daily basis.
Wild.
Way more.
Wild. Yes. Okay, so let's talk a little bit about gross examination or grossing.
Yeah.
Is how it's sometimes referred to. So this is the combination of examination, description, and sectioning of specimens. So specimens like tissue or organs before they're processed further. And so these samples oftentimes, I'm sure AJ has seen a range of scenarios, but oftentimes I would think they're from, surgery or maybe a biopsy.
Yeah.
So there's a lot of assessing, there's a lot of dissecting, and there's a lot of measuring of things. And it's really important. Grossing is really, really important because it's what allows us to get good diagnoses. Humans have been grossing stuff for a really, really long time. Lots of autopsies to better understand anatomy, like what's connected to what.
Right.
If someone dies, where are you seeing, if you're looking internally, are there certain structural changes or other physical changes, that kind of thing.
I just had the weirdest thought. Can you imagine being the first human to look inside the body of another human and just being
crazy? Because then you're
horrified?
Wait, is this what's in me?
Right. Yeah.
Right. I mean, I feel what came first, war or the first grossing exercise? Right? Because
I guess at the very least, people were hunting, right? So you know that there are the inside of an animal looks like.
Right. But actually sitting there and really dissecting things and looking at it must have been wild. Yeah.
Yeah, I mean, especially when you think about the amount of work that went into in Egypt, creating mummies and stuff.
Right.
It's that's they're working with the inside of a person's bodies and also figuring out what to do with the inside.
Right. How do you preserve, preserving the lungs versus the brain, very different thing, right?
Yeah.
Oh my gosh, so interesting. Okay. So grossing became more important to medicine in the 19th century when a man named Rudolf Virchow, I think I'm saying that correctly, Rudolf Virchow. So he was a German physician And I saw him described as the father of modern pathology. So he began teaching the importance of getting macroscopic, i.e. gross, descriptions alongside the microscopic ones. So things that you might see in the diseased tissue, only under a microscope. And then, of course, over the 20th century, so in the 1900s, there were a lot more advancements that were introduced. Things like using formalin as a tissue preservative. So that was the other thing. Like, you only had a certain window of time to really be able to look at these specimens before they would degrade.
Right.
And so finding the appropriate preservatives was also a challenge. And I'm not going to get into that, but like, that's really interesting. And I think about the work I did and using formalin, using formaldehyde to actually preserve tissue specimens.
Right.
Okay, so 1950s and 60s, there are more guidelines that are around specimen handling. There are actually steps. It's less of a, I don't want to call it a free-for-all, but I'm sure in some contexts it truly was a free-for-all.
Yeah.
So some important steps include examining visible aspects of the sample. So looking at size, shape, color abnormalities, taking pictures, because you can take pictures, right? Like, I mean, there's a lot of incredible anatomy drawings that were done. Just like the degree of detail is unbelievable.
Right.
But being able to take photos, easier,
for sure,
more accurate in what you're seeing, right? It takes out a bit of creative freedom there.
Though the photographers might disagree.
Yeah, sorry photographers, but anyway, so yeah, like another part of the process was cutting areas of interest for further processing. So looking at lymph nodes, looking at lesions. And then in processing, part of that would be draining the tissue of water, putting in a fixative. So that's where you talk about things like formalin. And then embedding in, say, paraffin wax so that you can then section it. These are really, really fine sections. I remember sectioning mouse brain that had been fixed and paraffin embedded, and like you're talking microns, right? Like it's really small. You're not even getting to a millimeter.
Right.
And then of course today, you know, you can use different staining agents, things to look at, different proteins that might be in the tissue, etc. Deboki, did you do much of that in grad school or undergrad?
No, the largest that I dealt with was like a cell.
Right.
I worked in a sea urchin lab my freshman year of college, but I wasn't really working with the sea urchins themselves, and mostly like we were looking at embryo development anyway.
Right. I mean, they're really cool.
Yeah, developmental biology.
This is going to get off topic, but like actually looking at like embryo formation in real time, you can actually see it form like under a microscope, which is wild.
Yeah, exactly. But I wasn't really doing much there. And then in grad school, everything I did was with cells. And so I think I just was a little bit shy of ever working with animals. Like I never got to that point.
Yeah.
And so I never did anything that would end up requiring it. I'm so curious because you're talking about like taking those tiny slices. Like how do you do that? Like how do you take such a thin slice? Like what are the tools?
There's a machine that helps you like essentially it looks like a deli slicer.
Oh my God.
A little bit. That blade though is so sharp. You have to be so careful. Like
I bet.
You barely touch it and it will slice you deep.
Oh my God.
And yeah, it's not good. Like
that's so scary.
I did not cut myself, but I was definitely present when people cut themselves and it was not great.
Yeah.
You got to be careful with that.
Right. Because it's like both the risk of cutting yourself and also contamination of whatever you're working with.
Right. Yeah. So you got to be really careful. But it's essentially like a little deli slicer with just an unbelievably fine, sharp blade.
Wow.
Yeah.
Oh my God.
Yeah. Okay. So Deboki, you found a couple of articles that are, more targeted toward people who are already somewhat familiar with grossing, but they're pretty fun. So one is an article titled The Art of the Biopsy, Finding Beauty in the Grossest Room in the Hospital. And the other one is more of a blog post from Kurt Schaberg, who's a medical doctor and a anatomic pathologist at UC Davis. And the blog post is titled Kurt's Grossing Basics. Kurt starts it off by saying, I remember how stressful and intimidating learning to properly gross a specimen can be. So with these guidelines and sample dictations, I hope to help those beginning this process learn the basics and have good technique. It's really interesting.
Yeah.
Definitely worth a read if you want to learn more about how people go about this and probably, I would assume, how AJ does some of their job. So that's kind of the like grossing background.
Right.
But then, Deboki, you got a little sidetracked doing this research, which we love.
Sure did.
Which we love. And you started diving into Rudolf Virchow.
Yeah.
So the guy who really made pathology engrossing a thing.
Yeah.
Who I mentioned I had seen described as the father of modern pathology.
Right.
He has a pretty wild bio, and I'm going to read some highlights, and I want you to hop in wherever you would like.
Yeah, I'm sure I will.
Yeah, so he said, quote, medicine is a social science, and politics is nothing else but medicine on a large scale. Okay, so he's a philosopher.
But like basically like a huge public health guy. Like
right.
That's like really one of his major contributions, along with being the father apparently of modern pathology.
Totally. So I read this and I was like, I need to stop and think about this for a second. But yes.
Right.
And then supposedly, this was so weird.
Yeah.
Otto von Bismarck. So I didn't know anything about this guy.
Oh, someone does not remember their APMEH.
Apparently, I don't.
Because this is like, I think he's like the only thing I remember from modern European history, like our class in high school was Bismarck was the guy.
Okay, yeah. So he was a diplomat who oversaw the unification of Germany and was its first chancellor from 1871 to 1890. And apparently, he challenged Rudolf Virchow to a duel. And the rumor is that Virchow decided to fight him with sausages. But you did note that, like, this probably isn't true, but it's really weird that this has become a rumor.
And it gets even weirder because, like, the fake story, I don't know if it's a fake story, but the story, the legend, the myth is that supposedly because Virchow was the one who got challenged, I guess dueling etiquette is that he got to pick out the weapon. And so he picked 2 sausages, one of which was edible, and the other was filled with roundworm.
Nice.
And then Bismarck refused the duel, and they ended up not fighting. Again, probably not true. At least the sausage part, I think, is not true. The duel might still be true. I'm not sure, but like,
right.
I think the idea that you could be a person, that this is a story about you, is already
it says a lot.
Wild.
Yeah.
Yeah.
No, totally. Like, you wouldn't have some, like, ho-hum dude have this story about him. So there's something going on. And in fact, he did some research on the roundworm.
Right.
Trichinella spiralis. And he was the guy who apparently established that we should be doing meat inspections. And there's a reason for that. Trichinella spiralis, or Trichinella spiralis, is a parasitic nematode. So it's a roundworm, and it causes trichinosis. So people get it by eating raw or undercooked meat, commonly pork, wild boar, or bear was what I saw listed. And I was like, never had bear, don't plan on eating bear. I'm sure there was more bear eating in Germany in the late 1800s.
Right.
So the issue is that it can contain that roundworm larvae.
Mm-hmm.
And ultimately, these worms will make it to striated muscle tissue, and they're going to burrow in. They cause a lot of issues depending on what muscle tissue they get to. Like, they could cause heart issues. They can cause breathing issues. It's really bad. Glad we do meat inspections.
Yeah.
But also, like, this guy was very pro-science, but he also was like, he didn't believe in evolution. He apparently called Darwin an ignoramus. To be fair, the concept of evolution was quite new at that point.
Yeah.
So I understand there was some pushback. And now it's like, what do you mean you didn't? But, you know, so I don't hold that against him a lot.
Yeah. He also didn't believe in the germ theory of disease, which is also fascinating. But it's also related in part, from what I understand, to him being so like public health focused. Like, that was a way that he understood disease was like an issue of like poverty and of like how we treat other people.
Yeah.
Like, he clearly cared about disease, and he just didn't believe
that was the route.
Pasteur's idea of like germs being the cause of it.
Right. I mean, like, you have to give a little bit of a pass for some of this stuff because this is all so new.
Right.
He was also like one of the first scientists to realize that cancer originated from normal cells, right?
Yeah.
Like, that's really cool.
Right.
And he was apparently the first to use hair analysis in criminal investigations. So, like, my question is, dude, who were you?
Yeah.
He had a lot of thoughts and feelings. Some of them were wrong. But yeah, this is a time before antibiotics. It's a time before vaccines. Like, people are just starting to understand things. Maybe he had worms in his brain. Like, I have no idea.
If you're the Rudolf Virchow biographer, let us know.
Oh my
we gotta know.
If you are the biographer, we need to do a full episode on this, dude.
Yeah, we will talk to you.
I am fascinated. We will 100% talk to you. So this was fascinating. Thank you so much, AJ, for writing in and sharing what you do for work.
And for doing the grossing.
Yeah, thank you for doing the grossing.
Yeah. Well, Sam, I've got a message from listener Tammy who writes, "Hello, Tiny Matters. Happy belated 100th episode."
Thank you.
I've been experimenting with sourdough and pine needle soda, among other things, and I'm fascinated with natural yeasts and how different they can be. What is yeast and where does it come from? How can the spore survive in store-bought flour, among other things? How do the different types of yeast and where they're found alter the taste? Which version should you use when? Why do some make carbonation and some make alcohol? I'm so fascinated by this world I didn't even know existed.
Yeah, yeast are cool.
They're really cool.
I don't know if it's less so now, but like even when I was in grad school, 8 years ago, a lot of people were still using yeast as their model organism.
Oh, yeah, totally.
Yeah, you can learn a lot.
I shared a lab space with a yeast lab, and so it was awesome because it smelled like beer and bread all the time.
Yeah, actually, that's really pleasant.
Yeah, it's great. So, Tammy, I don't know if I'm going to be able to answer all of your questions, but I think hopefully what we can get through today is sort of the understanding of why we have different types of yeasts. So first, some basic facts about yeast. They are fungi, but they look different from a lot of other fungi in that, I think a lot of times when we think of fungi, they, are branching. When you think of mushrooms, you think of them as, coming off of these, fungal networks that are all branched out and hyphaed out. But yeast aren't like that. They're usually kind of just, little cells, and they reproduce asexually through budding. So sometimes you'll see, two little yeasties, bound together, and that's just them kind of going through their process to
Two little yeasties.
Yeah, exactly. So the most famous species is probably Saccharomyces cerevisiae, which is usually called brewer's yeast or baker's yeast. In science, as we just talked about, it's really a commonly used organism for research for a lot of reasons. We know a lot about their genome. They're easy to grow. Same thing that makes them great for cooking also makes them great for science.
Yeah.
So we know a lot about yeast already from using them for the food and the drink. And now that makes them easier to use for science as well. And the way that yeast usually works in our food is through fermentation. So basically, while yeast usually use oxygen to break down sugar and make energy, it can also turn to alcoholic fermentation when oxygen is low. And some of the byproducts of that process include ethanol and carbon dioxide. And so when we're making bread, that carbon dioxide gets trapped in the dough. And so that carbon dioxide will then lead to the bread rising. So that's important for bread making. Meanwhile, ethanol is going to give you flavor and aroma. So that might show up in the bread. And then, of course, if you're making wine or beer, that's going to give you alcohol. That's going to give you
that's the goal.
Bits of buzz. Yeah.
I mean, among other things, but
yeah.
A lot of times that's the goal.
Yeah. And so there are different types of yeast. There's bread yeast, there's wine yeast, there's brewer yeast. And from what I can understand, I'm pretty sure they're all Saccharomyces cerevisiae with a little bit of some exception that we'll get into in a bit. And so there are some key traits, though, that are going to separate them. So even though they're all Saccharomyces cerevisiae, there are different strains. In the same way you might have different breeds of dog, but they're all the same species. You can have the same thing with yeast where you have different strains, but they're all still the same species. And so bread yeast generally has a lower alcohol tolerance compared to wine yeast. So basically alcohol, a little bit toxic to yeast, but some yeast are going to be able to survive higher concentrations of alcohol than others. So bread yeast can handle around 6 to 8% alcohol, while wine yeast can get up to 17%. Brewer's yeast, which you're going to use for beer, is more in the middle. It's around 10 to 12%. Then also wine yeast produces gas more slowly during fermentation, while bread yeast produces more gas and it's faster. And so that's great because bread yeast is something that you're doing in this quick, hot process. Right. So you get that really
You
fast
want that
process.
bread to rise. You're
exactly.
We need it to rise right now.
Yeah, you need it to be active and doing that thing.
Yeah.
Whereas wine, that process is a lot slower. You're doing it in barrels. It's not going to be as hot. So it's a longer process. So having that slower fermentation works really well for wine. And there are some like other differences as well, but those are the ones that I think are kind of the best, like most important things for understanding how these different types of yeast like make for different types of food and drink. But I was also really curious about whether all of these yeasts are Saccharomyces cerevisiae. And again, I'm pretty sure for the most part that they are, but I did find a paper that was talking about using yeast in wine production. And while the most common yeast species for wine fermentation is Saccharomyces cerevisiae, there are also some non-Saccharomyces yeasts that are used to make wine. And they're particularly important because they make a lot of the aroma and flavor compounds that are going to add to the complexity of wine. They're generally less tolerant of alcohol, though, so they're not as good for fermentation. They're still like important for like making the wine, but that's just like not their main purpose. They're going to be used usually in addition to Saccharomyces cerevisiae to give that additional flavor. And yeah, I think it's just really cool because like there's a whole separate conversation that we could have that we're not going to do right now, but that I've like looked into in the past about like this idea of whether we like domesticated yeast, because like originally they're like out there, like we found yeast because like they are sometimes just like out there fermenting fruits in the wild. Like they're just like on the fruit, and like sometimes you'll get that sort of like alcohol-y kind of smell with your fruit because of the yeast that are like just naturally occurring there.
Yeah.
And we, before we even knew yeast were there, we were like cultivating yeast to make foods, which is just wild. It's so cool to think about.
Yeah.
There's like so much more that now that we know about yeast, like I think we've been able to like further refine, but it's really neat to think about.
It is really neat. I have a business idea.
Yes.
And that business idea is I would love if there were a wine bar or something where the focus was the yeast. Where it's like, okay, so this wine was made using this strain of yeast or this other strain of yeast. And this is like, for me, I think I would find it so much more fun to try different wines if I knew some of the underlying like science or, you know, like the organisms involved. I think that would be really cool.
I bet there's a bar in like Cambridge and like in Boston that does this. There's got to be one.
There has got to be, but that kind of thing would be really fun to me where it's like, because I'm like, okay, yeah, I can like drink some wine and I can say, oh, it tastes kind of fruity or this or that. But I think that I find when I've gone to, not that I've been to that many vineyards or wine bars really, but I find that like it's more here's what you can sense. You know, here are the differences between this red versus this red, and it's about the grapes, but it'd be really fun to have it be about the yeast.
And I think that's like also what's really cool about wine. And I don't know how much we know about this, but I think this is sort of like an element of what makes like different regions' wines also taste different. Like there's so many things. There's weather, there's a lot of aspects. So I don't want to attribute it to just the yeast, but like I imagine there are like different yeast in different regions that maybe we know, maybe we don't know, that are shaping the way that these fruits are fermenting and like creating that final flavor, which is so cool to think about.
It's really, really cool to think about. Yeah.
Yeah. Microbes, man. They just do it all.
Microbes. We love them.
Yeah.
Can't live with them, can't live without them. No, we can live with them, so that's not accurate.
Yeah,
but
we have to live with them.
Can't live without them, for sure.
Yeah, apparently. Yeah. Well, thank you to AJ and Tammy for submitting to Tiny Show and Tell Us, a bonus series from Tiny Matters, created by the American Chemical Society and produced by Multitude.
You can send us an email to be featured in a future Tiny Show and Tell Us episode at tinymatters@acs.org, or you can fill out the form that's linked in the episode description. And I'll see you next time.